Hello and welcome to today's webinar from Physiomics, presented by Hitesh Mistry on Virtual Tumour Clinical. Today we will go through an introduction to Physiomics, followed by an introduction to Virtual Tumour Clinical, which is a mathematical model describing the growth of a tumour, and we will then present emerging case studies on Virtual Tumour Clinical within the metastatic melanoma setting. These case studies will involve 3 drugs, Vemurafenib, Docetaxel and Selumetinib. So firstly, an introduction to Physiomics. So the business was founded in 2001 and listed on the AIM stock market in 2004. We are based in Oxford within the UK. Physiomics' main focus has been on developing predictive models to try and improve the success rate of drug discovery and development projects, with the sole purpose of reducing timelines, saving money, which eventually would lead to better decisions. So it's all about sort of reducing the total number of experiments you have to do through the discovery and development sort of timeline. Our particular focus has been in oncology, and today we'll take you through our unique mathematical model of a growing tumour called virtual tumour. Physiomics has an extensive track record on successfully completing projects, with a total of 45 completed so far. We have 3 large pharma customers. Lilly were one of the ones that first licensed the progenitor version of the virtual tumour platform. We have 2 other pharma companies, one top 5, who are actively engaged in the preclinical virtual tumour projects, and both of these companies have expressed an interest in looking at virtual tumour clinical. We have several biotech customers, specialty pharma and other customers. We are also being quite successful within obtaining grants and delivering on these grants from numerous awarding bodies. Virtual Tumour Clinical has been in development for nearly 2 years now and has been gathering pace. We have 2 academic collaborations, one with the NIH and one with the University of Oxford, and today we'll be taking you through the work we've been doing with the University of Oxford. We also have 2 large companies who have been in discussions over a significant VT clinical programs, and we had our first commercial customer on VT clinical back in 2013. We also have a few other products that are in development, basically a cardiotoxicity model and drug card database, which will launch late in 2013. For those products, please see other webinars which we have made available hopefully through our website soon. So today's talk is all about the virtual tumour. And what I'll do first of all is take you through what the virtual tumour actually is, what data is required, and what we actually produce as predictions. So the virtual tumour is a mathematical model that describes the growth of a tumour, basically an individual lesion on the back of a mouse. Originally it was designed to look at xenograft studies. So the virtual tumour, the mathematical model, is able to distinguish between the viable rim and the necrotic core. So it takes into account the spatial structure of a growing tumour. The unit of analysis is the cell, and this allows us to look at sort of synchronization and desynchronization. And the synchronization effects can be through to a direct or indirect effect on the cell cycle. So each cell represents basically a state within a cell cycle at a given point in time. The typical data that we use is basically xenograft growth data. So this is basically tumor volume over time. So we would have control data, dose response of drug A, a dose response of drug B. We'd have the PK data to go with those dose response data. And then we would make predictions regarding the combination schedules, usually relying purely on the monotherapy data. So this would be changing the doses, changing the sequence, changing the times, changing the off days between 2 drugs, off times between 2 drugs. And this relies basically on our unique virtual tumour model. We also take into consideration certain PD data, so for example target of mechanism data, and certain other biomarker data that involves looking at cell fate are also quite important. So cell death, whether or not the drug does have an indirect or direct effect on the cell cycle is also quite important. So that tells you a little bit about the data, and so what I'm now going to show is, hopefully with my arrow, is also highlight what it is that we actually do. So what you're seeing here on the right hand side, on the x-axis is time in days, and on the y-axis is the tumour volume in cubic millimeters. So once we've received dose response information for drug A and dose response information for drug B for the Xenograft studies, we are asked to make predictions around the combination schedules. So, what order would you give these drugs in, what doses, time between doses. And what we produce basically is a mean prediction, which is this red line, and then a confidence interval, which is the pink shaded region. So we provide these predictions up front before the experiments have been conducted. And then after the experiments have been done, our customers usually overlay the predictions on top of, overlay the experimental results on top of the predictions. And that's represented by this black solid line, which represents the mean. In this case, was the Xenograft experiments, the mean of a cohort of mice, and then the standard errors are given by these vertical lines. You can see the individual mice behavior as well. So here is an example of where we made two predictions up front before experiments had been made, and then once we received the experimental results, we can see that we were accurately predicted the effects of these two different regimens. So basically, in a nutshell, the virtual tumour is basically a mathematical model of a growing tumour. It's able to distinguish between the viable rim and the necrotic core. Its unit of analysis is the single cell, and each cell basically has an instance of the cell cycle running, such that we can then look at synchronization and desynchronization effects, both direct and indirect effects of drugs on the cell cycle. Right, so won't go too much into detail on the preclinical virtual tumour. We already have a webinar for that, which you should hopefully be able to find the links of through our webpage. And just to recap on the preclinical virtual tumour, is that it predicts the change in mean tumour volume over time. That's what we saw on the previous slide. We have completed nearly 35 preclinical studies which have confirmed the predictivity of the model. Like I said, the model describes the growth of a single tumour, i.e. a single lesion on the back of a mouse. In the clinic, we've decided we'll stick with the same sort of methodology, i.e. we will try and predict the change in mean tumour diameter over time for all lesions in a certain arm of a study. So why are we going for predicting the mean behaviour? Well, we know from the preclinical work that the mean behaviour is predictable, and we decided to stick with this as we go into the clinic initially. And the other thing we've looked at is that we would like to look at the sort of individual lesions within patients rather than some sort of metric that describes the overall status of tumour burden within a patient, i.e. sum of longest diameters. And the reason for this is we want to compare like with like. So in the mice, we're looking at a single lesion, and we're making a decision there on whether or not to take a drug into the clinic based on what happens to the mean behaviour on a single lesion. So we tried to say in the translational space, we'll look at exactly the same metric in patients. We will look at the mean change in lesion size over time, over all lesions in different arms of the studies. So typically when you move from preclinical to clinical setting, and vice versa, the current pharma approach simply involves matching the pharmacokinetic profile between xenografts and man. What they do not take into consideration are the different growth dynamics. And hence, this is one of the reasons why people started to look at patient-derived xenografts, as they realize that the growth of human tumors is actually far slower than what you see in xenografts. So if we think about xenograft volume doubling times, we're talking on the order of days, whereas in humans, a volume doubling time could be weeks to months to years. So clearly there's a difference in growth dynamics, and taking this into consideration is we believe is quite important if you want to make translational predictions. So the objective of virtual tumour clinical is be able to sort of say, well, from the preclinical mathematical model, can we just adjust certain key parameters that then translate the growth effects of preclinical tumours to clinical tumours, with the hope that then we should be able to make accurate predictions about drug effects in man based purely on xenograft data and limited PK data in man. So now on to virtual tumour clinical model development. So as I mentioned earlier, one of the main aspects of the virtual tumour is that it's able to distinguish between the necrotic core and the viable rim. So what's been important to us is to look at the differences between these sorts of parameters in the preclinical space and in human tumours. So we've been looking in the literature at the viable size of viable rims, how thick are the viable rims in the clinic across different tumour types versus what you see in xenografts. What percentage of cells within these viable rims are proliferating by looking at Ki67 data from literature, what percentage are apoptotic by looking at PBCAS-based 3 data. And we're also generally interested in the growth of clinical tumours. We know it's quite variable in the clinic, but we're also interested in what's that variability like in different disease areas between, for example, breast and prostate, breast and lung and prostate, etc. The other thing that we're quite interested in is understanding, well, how quickly can a tumour shrink? So given if you have a drug that actually works, how quickly can an individual lesion shrink when a drug is applied in human tumours? Now, this is quite important to a metric that's not really looked at too much within the literature. And this is one of the reasons why we did the first case study, was to try and get a feeling for, within a disease area, well, how quickly can a lesion shrink? The other aspect that's different between preclinical and clinical tumours that we thought would be important for us is looking at the variability in durations of cell cycle phases. Now, I'm well aware there's many other differences we can look at, for example, genetic heterogeneity and so forth. But our main focus here initially were on things that we thought would help us with this idea of predicting the mean behaviour. Clearly, genetic status and gene expression data will probably help with understanding how the variants would behave in the clinical population. But our focus here initially was on predicting mean behaviour. So in order to answer one of the questions around about, well, how quickly can a lesion shrink, the first thing we started to look at was basically a back translational project. So here we're basically going to ask the question, if we were to calibrate virtual tumour to clinical data, are we then able to sort of make predictions around what would happen in a preclinical setting based on the fact that we'd basically calibrated the model purely to clinical data? And in this, basically, we were looking at the metastatic melanoma setting. So the drug of interest is Vemurafenib. It was approved by the FDA in August 2011. It caused an increase in overall survival. There were high response rates, and there's plenty of work going on looking at combinations of this drug with other agents within the metastatic melanoma setting. So Vemurafenib is a BRAF inhibitor. Like I said, it's used within the metastatic melanoma setting for patients who have the BRAF V600 mutation. So in order to calibrate virtual tumour clinical, we need clinical data. The clinical data we have are basically 20 patients where each lesion was monitored over time for a certain dose of Vemurafenib, the actual approved dose. But remember, what we're interested in, as I said at the beginning, is that we're looking at what happens to the lesions over time, not necessarily the number of patients. So what's important to us, the important end value, is the total number of evaluable lesions. In this case, we had 69 lesions. So we have the shrinkage data from 69 lesions. We also need a PK model, a pharmacokinetic model, and this is contained within the FDA report, which is what we used for our PK/PD model. Now, in order to be able to do a back translation, we need to have some data that we can actually test our model against. And for this, we had to go and source some preclinical data. And interestingly, the only sort of article where we saw dose response within the preclinical setting was involving using a Colo-205 xenograft. So this is a colorectal cell line which happens to harbour the BRAF V600 mutation, which people believe is important to look at Vemurafenib efficacy. Now, even though Colo-205 represents a colorectal cell line, we believe what's more important is actually the mutation status of the cell line, or even more so than mutation status is the pathway that the cell line relies on for the tumour to grow, than mutation status or the actual tissue type. So we were quite comfortable here believing that hopefully we'd be able to make a prediction into this cell line model, mainly because this cell line model relies on the signaling axis that Vemurafenib was designed to inhibit, to proliferate and to grow as a tumour. Again, for the preclinical data, we had to source a PK model. And again, this is a well-studied drug, so there's plenty of options there in the literature for us. From the wide range of literature evidence that we had, it's quite clear what the mechanism of action is for Vemurafenib. It's known to exert its anti-tumour effect through causing G1 arrest. So we're mainly dealing with a cytostatic drug here. So we'll now go through basically what our modeling plan was. This is a step-by-step guide of exactly what we did. So the first step is to basically analyze the clinical data using a standard population analysis approach, which is basically a mixed effects modeling approach or hierarchical modeling approach. And like I said, our goal is to be able to predict mean behaviors. So we then calibrate the virtual tumor to the mean clinical signal. What we then do is we sort of say we're going to switch the growth settings. So we're going to take out the system parameters which describe clinical growth and swap them back in for preclinical growth settings that we've been using successfully for over 35 projects. We then calibrate the preclinical model only to the growth of the control xenograft. Now this is to make sure we've got the cell cycle time right for the preclinical setting. We then swap out the clinical PK for the preclinical PK, and then we make a prediction regarding the preclinical monotherapy effects. And then we will there compare the prediction with the actual results. So one way to look at this is a little bit like the PBPK approach, where you would swap in and out certain parameters that represent different species. So what we've been trying to do with virtual tumor is to have a set of parameters that represent xenograft growth, and then sets of parameters that represent different types of clinical growth. So from breast cancer to prostate to lung to metastatic melanoma, etc. So on this slide you're seeing steps 1 and 2. So this involves clinical calibration. So again, I'll get my green arrow and try and explain to you what the axes are and what exactly we did here. So on the x-axis here we have time in days. On the y-axis we have the diameter in millimeters of each lesion, and each lesion is represented by a series of circular dots which are joined together by a line. So this basically describes the time course of an individual lesion when a drug is applied. Now what you're also seeing here is the results of a population analysis. So this is sort of saying a mixed effects analysis, where we're basically asking, well, what is the mean behavior from these sort of cohort of lesions? And what you're seeing here is the black line represents the mean of the population analysis, and the dashed line represents the 95% confidence intervals. And then what we've done then is basically calibrated the clinical virtual tumor to this mean line. So no surprise, this red line, which represents the mean of our virtual tumor, sits right on top of the black line. So no surprises there, and we have a confidence interval as well. So once we've calibrated the model to the clinical data, we then swap out the growth settings from the clinical setting to the preclinical setting. So these are what we call system parameters. They're not related to the drug at all. The drug potency parameters are only ever calibrated from the clinical data. So we then calibrate the model to the control growth of our xenograft. So again, on the x-axis here we've got time in days, and the y-axis is tumor volume in cubic millimeters. And the black solid line represents the experimental data with the mean and standard error of the experimental data. And then the red solid line represents the calibration of our virtual tumor model, and the pink region basically the confidence interval. And remember, what we're doing here is calibrating the model. So this doesn't necessarily mean we'll have the best fit to the data, as we believe what's important is capturing the early growth dynamics, as later growth dynamics are usually can be subject to dropout effects of mice. And here we did not have the individual mice to help us out to understand, well, what were the dropout effects in terms of their effects on the mean behavior. So our focus is always on the earlier dynamics. So now we've calibrated the model to the preclinical control. We will now have a go at predicting what would happen to 2 different doses with Vemurafenib within this Xenograft study, with potency parameters estimated from clinical data. So these are the predictions, the monotherapy predictions for 2 doses. So the left panel represents the 6 mg per kg dose given once daily, and the right hand side panel represents the 20 mg per kg once daily dose. So these were our actual predictions. So how well did we do? We now overlay the experimental results, and we can see that the virtual tumor model was able to capture the experimental data quite well. So you can see here that we were able to capture the growth dynamics early on quite well, but then we do see this drop in mean effect, which we believe is down to dropout effects of the mice. But this gives us quite good confidence that we have pretty good ideas of how to alter the growth settings between the clinical and preclinical situation, as we were able to accurately predict the effect of the preclinical xenograft experiment here. On the right-hand side panel, we were able to sort of suggest that this is the 20 mg per kg dose would give you more or less tumor stasis, which is what you were seeing in the experiment. It's not a complete exact sort of prediction in terms of we don't have complete overlay on top of the experiment and the model, but we are able to definitely suggest that you would see tumor stasis here, which is what we're seeing in the experiment. So to summarize this first part of the project, we were able to calibrate the virtual tumor to monotherapy changes in clinical lesions. And by doing this, when we calibrated the mean behavior in the clinical population, we then made a prediction on what would happen in the preclinical xenograft studies, and we were able to do this accurately, capture the preclinical dynamics very well. So just to recap exactly what we did here, we calibrated the virtual tumor to the clinical data. We then swapped out the clinical growth settings, which are system parameters, for the preclinical growth settings, so the potency estimates stayed the same in the clinical and preclinical setting. We then calibrated the preclinical model to the control data, and we then made predictions regarding the 2 doses in xenograft studies in the preclinical setting, and we got them correct. So this was basically what we'd call a successful back translational validation. Now, obviously, people don't really care too much about, well, can you predict the effect of a xenograft based on what's happened in man already? We would actually like to do this in a forward way, based on preclinical data and limited clinical PK data. Can we make a prediction about what would happen in a patient cohort in a forward translation, which is what we will now look at. And just a quick aside here before we move on to that aspect of the project. Remember I said that we were working in a metastatic melanoma setting for making predictions in a colorectal cancer xenograft. Now this might sound a bit crazy to some people, but then if we think about it, what's really important is what's driving the cell growth, the tumor growth. As long as the drug has, as long as the pathway that's important for driving preclinical tumor growth is similar to that what's in patients, then the tissue type is not necessarily important. And also we hypothesize that the mutation status isn't all that important, which we'll see in the next project. So now we will go on to doing a preclinical to clinical translation, and again in metastatic melanoma. And the reason why we chose to stay in metastatic melanoma is because we have this back translational information which gave us an idea of, well, how quickly can a metastatic melanoma sort of tumor shrink? How does it grow? What xenograft can we use to translate? So we had all this prior information, which is quite important, we believe, to making a forward translation. This was a case study done with Mark Middleton at the Oxford Experimental Cancer Medicine Centre. This case study we entitled Translational Qualification of Virtual Tumour. So this was to determine whether our technology could accurately make a forward prediction in basically predicting the mean change in tumour size over time in a 2-arm phase 2 clinical study. The clinical study involved looking at Docetaxel plus placebo versus Docetaxel plus Selumetinib in BRAF wild-type metastatic melanoma. Selumetinib is a MEK inhibitor currently is currently in clinical studies but from AstraZeneca. So what was our start point here? So what we had here in terms of preclinical data was single drug xenograft dose response data and preclinical and clinical PK. We did have some combination xenograft data which we used as a sanity case, but it wasn't actually needed to make the predictions that we made. So duration in this project took roughly 6 weeks because all the data was actually available. The outcomes, well, we were able to correctly predict the mean change in tumour size over time in both arms of the study. And we also then subsequently have been providing sort of new options in terms of schedules to try and ameliorate the toxicities seen with the current existing schedule. So the Docetaxel-Selumetinib clinical study, it involved 40 patients in each arm roughly. And like I said before, what's important to us is the number of lesions. And we have approximately 100 lesions in each arm. So this was the BRAF wild type setting. So this is different to the previous setting where we're looking at BRAF mutant setting. But the belief here was that, again, if you're applying a MEK inhibitor within this setting, you're assuming that the MAP kinase signaling is actually driving the tumor growth in this patient population. And with the colorectal cancer cell line model that we looked at before, even though it harbored a BRAF mutation, that BRAF mutation basically meant that the MAP kinase signaling pathway was active. So the MAP kinase signaling pathway is active in both our Xenograft and, we believe, in this patient population. Like I said before, selumetinib is a MEK inhibitor currently being investigated in numerous disease areas. There's also other MEK inhibitors out there, and the recent one is the trametinib by GSK, which is approved last year in the BRAF mutant setting within metastatic melanoma. Now, in order to be able to do this sort of forward translational project, what we were looking for in the literature was preclinical xenograft data involving this colorectal cancer cell line, which we thought was important, preclinical PK data, and then also some clinical PK data to help us make these predictions in this clinical study. So we were able to source all these things, and we'll just briefly go over the xenograft data and what we did in terms of our modeling plan, and then we'll show you the predictions. So as you saw before, we have our step-by-step guide to our modeling plan. So in step 1, we basically calibrated the virtual tumor to the preclinical data for each agent. This involved using literature PK and xenograft data. We then swap out the growth settings, the system parameters, not the potency parameters. So the system parameters describe the preclinical growth for the clinical growth settings that we'd ascertained from our back translational project involving Vemurafenib. We then plug in the clinical PK and then make predictions regarding the mean behavior in each arm of the phase 2 study. We then perform a population analysis of the clinical study and then compare our prediction with the actual result. So step 1 involves the preclinical calibration of the MEK inhibitor. So we only found one paper where we found a dose response for this MEK inhibitor, and what you're seeing here is a calibration of virtual tumor to this dose response data from the literature. So I'll get my little green arrow. And again, what you're seeing here on the x-axis is time in days, on the y-axis is tumor volume in cubic millimeters. This is the calibration of virtual tumor to the control data. So again, virtual tumor is the red line and the pink region is the confidence intervals, and the black line represents the mean behavior from the experimental study with standard errors. And that was the control. That was then the low dose, which I believe is 0.5 mg per kg. This is the 2.5 mg per kg, and this is the 25 mg per kg. Now, as you can see here, we were able to calibrate the model quite well to this dose response data. So no surprise, we're making a calibration, we're not making a prediction here. So then we also needed data on docetaxel, because remember we're trying to predict the combination behavior here. So we need some information on docetaxel. We worked internally with docetaxel quite a lot, so we understand its accumulation. This is PK accumulation in the tumor. We have a fairly good understanding of the mechanism of action. And so what you're seeing here is basically the calibration to the control data that where we source the docetaxel information, and then the calibration to the 10 mg per kg dose within that xenograft experiment. Again, on the x-axis here we have time in days, on the y-axis is tumor volume in cubic millimeters, and the black line represents the experiment and the red line represents our model. So again, no surprises here, we're able to accurately calibrate the model to the preclinical experimental data. So one thing I forgot to mention is that we're assuming that the MEK inhibitor's mechanism of action is similar to Vemurafenib in that it causes a G1 arrest predominantly. So that's the calibration. So once we've calibrated the preclinical model to the drug data, so i.e. we made an estimate of our potency parameters, we swap out the system parameters, which is the preclinical growth settings, for the clinical growth settings. So we do not touch the potency parameters at all. We then replace the preclinical PK with the clinical PK and then simulate the clinical dosing schedule. And we then provide prediction intervals for these schedules, which is what you're seeing here. So on the left-hand side here, we have the Docetaxel, which was given 75 mg per square meter once every 3 weeks with placebo. On the right-hand side, we have Docetaxel given again 75 mg per square meter once every 3 weeks, together with Selumetinib twice daily with 75 mg. On the x-axis here, you're seeing time in days, and on the y-axis, you're seeing longest diameter. And then we provide this sort of prediction interval here from the virtual tumor. And our mean basically sits right in the middle of these prediction intervals. So now we've made the prediction using the virtual tumor. Let's see how well we did. And this slide shows the successful prediction of virtual tumor. So we'll take you through this a little bit step by step again. So once we've made the predictions, we basically do a population analysis of the clinical data and we overlay the results. So as previously mentioned, the experimental data here is represented by a single little circle, black circle, which are joined together, which represent the time course for an individual lesion. The solid black line basically represents the mean sort of result from the population analysis, and the dashed line represents the 95% confidence interval. And as you can see, our prediction sits right on top of the actual experimental result. Now, the actual result of this study was that there was a slight increase in response rates in the combination arm with selumetinib versus placebo. So I think there's approximately 35% response rate in the combination of docetaxel and selumetinib, compared to a 14% response rate in the Docetaxel placebo arm, and this had a p-value of about 0.059. So this basically, our model was able to capture this sort of mild treatment effect that they were seeing in the clinic through response rates. So one thing we could possibly look at here is to try and relate then virtual tumor to an actual clinical important metric such as response rates. But what main thing to look at here is that we were able to basically capture the mean behavior accurately, make a successful translational prediction using very limited preclinical information and limited clinical PK data. So just to go back a little bit and think about, well, what xenografts were we using here? Now, I said before that what we believed what was important is that the xenograft cell line that you use actually represents the pathway that you want to inhibit. So i.e., what do I mean by that? So that the xenograft grows basically dependent on a certain signaling pathway that you want to inhibit. So if, for example, here, both in the BRAF mutant clinical population and the BRAF wild type clinical population, we're assuming that the MAP kinase pathway is still driving this growth of clinical tumors. And the xenograft cell line that we're using has this effect. So the COLO205 has this BRAF mutation, and we believe that that means that basically it's driving the growth of the tumor through the MAP kinase signaling pathway, similar to what we're seeing in the clinic. Hence why I believe that the COLO205 model was a good cell line model here to use to make a forward translational prediction. Now, what's interesting here is that we did not need to use a patient-derived xenograft. And this brings on to some interesting questions on, well, with patient-derived xenografts, what are the pitfalls? So if you think about virtual tumor, we've been able to capture this sort of slower growth human tumors, which is what the purpose of a patient-derived xenograft is. So in some ways, you could say that actually the virtual tumor is a surrogate for a patient-derived xenograft. In some ways, it could be a better surrogate than a patient-derived xenograft. And the pitfalls of a patient-derived xenograft, we believe, is that actually it's a case of biological overfitting, because the microenvironment you've taken from the patient is unique to that patient. So how well can you extrapolate to a wider population, which is what you're being asked to do when you're designing clinical studies? And so therefore we believe that it's not such a an obvious thing to be able to do with a patient-derived xenograft. So a brief sort of recap on what the data requirements would be to do a virtual clinical project. And as you've seen through this presentation, what is quite important is having both preclinical and clinical data for a different drug in a similar setting to what predictions are required for the new drug. And the sort of data that we really require is basically PK information, PD, that's usually the mechanism of action of the drug, and basically the cell fate type of data, as well as xenograft volume data over time. And also what's important is trying to pick a xenograft that you believe is relevant to the patient population. In terms of clinical data, usually clinical PK profiles are fine to make a forward prediction, or predicted clinical PK is also useful. And then to make this a prediction, we will require the initial size of each lesion before treatment. The longest diameters are fine. So like I said, the ideal situation would be where you have some prior information within a certain disease setting on a different drug that we could then use to try and help us make a forward prediction. We're currently looking at other disease areas, so we've been looking at the same sort of project in prostate cancer and breast cancer. So just to summarize the entire presentation today, like I say, we did a back translational study first, looking at Vemurafenib, and then we did a forward translation involving Docetaxel and Selumetinib, and we showed that we were able to successfully predict the combination results using only monotherapy preclinical efficacy data and clinical PK data. We have now recently performed further predictions for the Oxford Experimental Cancer Medicine Centre, exploring different dosing regimens and swapping out Docetaxel for Paclitaxel within that setting. So I think you can see here, and it's quite clear that the cost savings that virtual tumour clinical can provide by first of all reducing the number of clinical studies you'd probably need to do to be able to find a schedule that will provide you with efficacy, to find you an optimal dose and hopefully an optimal schedule. And I think there's another cost saving aspect here, is that what we're saying is that you don't necessarily need as expensive patient-derived xenografts to be able to make these predictions. As so far, we have not relied on these to make these predictions, and we've done quite a successful job of making predictions without the need for these patient-derived xenografts. So overall, we believe that virtual tumour clinical will play a key role in sort of reducing attrition rates by speeding up, hopefully, the translational process, by hopefully providing customers with an answer to a dose and schedule as quickly as possible and far quicker than you can currently do. For further information about virtual tumour clinical, please feel free to email the company. You can email the CEO, who's Matt Chadwick, on this email address, and we also Chief Operating Officer Christoph Schasigmont. We will now take some questions, which will be answered privately through the dialog box. And I'd like to thank you today for listening to a virtual tumour clinical presentation that was presented to you by Hitesh Mistry. Thank you and goodbye.